Assalamualaikum and good morning. My name is Afiq Kamil Muhammad Shahizam, and today we will be doing a presentation about the determination of ofloxacin in urine using UV Vis absorption spectroelectrochemistry. Firstly, let me introduce you to the topic at hand. So, firstly, we want to find ofloxacin. Ofloxacin or OFL is a second generation fluoroquinolone FQ with a N-methyl pyrazine group attached to carbon 7. Large amount of OFL has been found in many aquatic environments according to research. 70 to 80% of OFL were excreted via urine to domestic wastewater and sewage treatment plants. The OFL contaminated water leads to the adverse effects to humans and ecosystem and also the environments. UV spectrometry and fluorescence are usually used to study OFL, but in this work, SEEC is used to develop a new method to quantify OFL in a simple and reproducible way. The objective of this work is to quantify OFL in a real urine sample using a new UV vis cell. Next, we take a look at the experimental section. So firstly, we have the materials and reagent. The materials are ofloxacin OFL and uric acid. So in this experiment, all solution were prepared in 0.1 mole KCl and 0.1 mole Britain Robinson or BR. The buffer solution contains boric acid, phosphoric acid, acetic acid, and sodium hydroxide. All solutions were prepared using high-quality ultra-pure deionized water, and the experiment were performed at room temperature. And secondly, we have the SEC. So the SEC that we'll be using is the UV Vis SEC, and they were done using SPELEC instrument controlled by DropView SPELEC software. Next, we take a look at the SEC cells. In finding the result needed, a new SEC cell was designed. In figure 1A, SEC cells were shown with two bodies with four 5 mm holes at each corner where magnets are placed to allow reproducible magnetic fixation. There's also pillars that have the alignment of optical fibers at different path lengths. There's also the circular cavity was designed to allow work in semi-infinite regime during SEC experiments. Next, we have the SEC setups. In this experiment, the light beam interrogates the solution in a parallel direction to the WE surface. To perform the SEC experiment, the SPE is first placed in the lower body and 50 microliter of test solution was added. The drop have to cover the three electrodes of the SPE. After, the upper body will be attached to the lower part using the magnets at each corner. Lastly, the SPE is connected to the potential tap. Potential.
Next, I am going to talk about the data analysis. Firstly, they are going to study about ofloxacin by using UV Vis SEC. Firstly, they are going to measure the cyclic voltammogram of ofloxacin. This is to measure the oxidation state and also to determine the energy needed for the molecule to gain or lose electrons. If we look at graph A right now, it is a CV graph, we can see the oxidation peaks at 0.99 volts. And if we look at graph B, which is the contour plot with spectra during the potential, the excitation happens at 245 nanometer and the emission happens at 294 nanometer. And graph C clearly shows what happens and at 294 nanometer, we can see a pi-pi transition, and at 245 we can see a n to pi transition, pi star transition. And if we look at the molecule of ofloxacin, it fits in with the data. And graph D shows the molecules. The result are almost the same, which means the ofloxacin can be determined this way. Next, we are going to study about the uric acid by using UV Vis SEC. We are going to measure it using they are going to measure it using the cyclic voltammogram. This is to measure the oxidation state, same reason as the afloxacin. Uric acid was measured here due to its prevalence as a matrix in urine, and this test was run as a reference for the simulation test, where a sample urine was spiked with both afloxacin and uric. This is to simulate a real urine. Now, if we look at the CV graph, we can see two absorption bands that decreases during the anodic scan. And the spectra shows a weak negative band at 237 nanometers and a more intense band at 286 nanometers. This band are related to the depletion of uric acid. Graph C is the CVA, shows a continuous decrease of absorbance starting at zero point 0.4 volts. And graph D, we can see the relationship of absorption at 286 nanometer, which has a similarity between the evolution of DVA and also the CV. Now we're going to talk about the simulated urine test. They prepared four types of sample: a sample of synthetic urine, a clean urine, a sample spiked with ofloxacin, a sample spiked with uric acid, and a sample spiked with both ofloxacin and uric acid. They are labeled as T1, the one without any, the clean one. T2, the one with spiked with ofloxacin. T3, the one spiked with uric acid, and T4, the urine spiked with both uric acid and ofloxacin. And if we look at the graph, for graph A, T1 stands for the urine, and if we look at T3, the one spiked with uric acid, the peak at 0.45 volts overlap each other, making it hard to quantify the contents of uric acid. But for T2, the one spiked with ofloxacin, the peak is only by itself, which is a good sign. And graph B shows the decrease of absorption band related to the oxidation state of uric acid. But here, graph B shows only the data for T1, which is the clean urine, which it peaks at 286 nanometer. While for graph C, the one spiked with ofloxacin, this is T2, the absorption band shifts to 291 nanometers. And graph D shows the oxidation of all the samples. And if you can see, the T1 goes straight line and goes back in a relatively same manner. And for T2, the green one, starts around the orange line and then goes down around 0.45 and then decrease, then goes back around without any deoxidization. And also, if we see T3, the one spiked with uric acid, has a major decrease at 0.45. And for T4, for both, it shows the oxidation, the combined oxidation line for all of them. Moving on to the repeatability of the test and the calibration model to determine the ofloxacin in urine. They found out the percentage relative standard deviation value at 1.1 volt is less than 5%, which is a good sign. The result demonstrates a well-defined spectroelectrochemical response, but sadly, the overlapping peaks of uric acid and ofloxacin absorption band makes it hard to quantify the ofloxacin using linear regression model. Therefore, they are going to employ parallel factor analysis, or PARAFAC for short, to measure the ofloxacin in sample urine. And they found out that the core consistency diagnostic, or cocondia, value was at 96.01%, which is close to 100%. If we look at graph A, the peaks at 286 nanometers, shows for component C, which is the ofloxacin. And for graph B, the calibration for the calibration curve also has a different curve than the others for between ofloxacin and the others. With the calibration curve plotted, the concentration of ofloxacin was determined. They found out the concentration was around 41.9 micromolar plus minus 2.5 micromolar. The accuracy was good and the recovery rate was astonishingly high, which is 98.1%, and the relative standard deviation percent was also good, which is 2.0%. This demonstrates the perfect model perfectly fulfills the objective, which is to determine the ofloxacin amount of ofloxacin in urine sample. If we look at table 2, which shows the parameters obtained during the determination of ofloxacin and uric acid in mixture, they found out the regression line for the R square line for ofloxacin was good, which is 0.9957. But for the uric acid, the R square value was quite low, which is 0.9625. But for the residual standard deviation, the ofloxacin has a lower value than the uric acid. And surprisingly, for the limit of detection, the ofloxacin has a lower LOD compared to uric acid. The uric acid has triple the amount of LOD compared to ofloxacin.
Lastly, we can conclude this presentation with the conclusion. So to conclude, using UV vis exclusion spectrochemistry, we were able to determine the OFL that can be found in urine. In the experiment, a new SEC cell was introduced to allow a more sensitive and reproducible quantification of OFL in urine. From the result, we conclude that the new SEC cell, with the new SEC cell and a good setup, a better method to quantify OFL was achieved. That is all from us. Thank you very much.